So hi everyone, and thank you firstly to Oxford Nanopore for inviting me to come and give a talk today. So I want to just kind of give you an overview of what we're doing in our lab and how we're applying and using Oxford Nanopore technologies. So our lab, we're particularly interested in looking at the regulatory regions in the genome and how these regulatory regions are controlling gene expression. So we're controlling when and where genes are being turned on and off. These regulatory regions are really essential to creating cellular diversity, particularly during development. So for creating brain cells, bone cells, but we have a particular interest in the pancreatic beta cell. The issue really is that we don't have a full understanding of how these regulatory regions are actually controlling gene expression. And it's really important that we do start to get an understanding of how this works because we know that variants within regulatory regions can result in disease. So these variants can either be the cause of disease or can contribute to disease. As I said, we're interested in the pancreatic beta cell. So I've given you two examples here which are relevant to that cell type. So firstly, an example where a variant in a regulatory region can cause disease is a single variant within can cause a disease such as congenital hyperinsulinism. On the flip side, variants which can contribute to disease, so multiple variants which contribute to disease, examples of this are shown in type 2 diabetes. Again, though, we really lack the tools to measure these sequence variants within these regulatory regions, which does limit our understanding of how these variants cause disease. So I wanted to give you kind of a motivation for why we want to look at these regulatory regions. So we recently found variants within the regulatory region of a gene called HK1. And variants in this region result in congenital hyperinsulinism, which is an overexpression of insulin. It's basically like the opposite of diabetes. And what we found was there was a high density of variants within this region, within a very short region. So we found 26 variants within a 50 base pair region. But what was interesting is that these variants defined 3 transcription factor binding regions. So our question then was, what combination of these transcription factors are binding to regulate this gene? And we know that multiple transcription factors binding simultaneously does control gene expression, and different combinations can provide specificity and control to gene expression. But what we really need to be able to do is measure multiple factors on the same DNA molecule. And traditional short-read sequencing methods don't really allow for us to measure multiple transcription factors on the same molecule. So this is where we kind of want to try and start to change the game a little bit. So we want to mark all of the accessible regions of the DNA. And this technique was alluded to yesterday by Philip. So the idea is that we want to, as I said, mark accessible regions. So we use exogenous methyltransferases to do this. So if we take, this is our DNA molecule here with our transcription factors bound onto it. Firstly, we use an Eco-GII methyltransferase, which marks all of our A's and T's depending on the strand, which we can then sequence. We then also use another methyltransferase, which marks all of our GC sites. So it's important to note that this is distinct from our endogenous CpG methylation. So the idea is then that we've marked all of our A's and all of our GCs that are accessible. But then we have this footprint where there's no methylation, where our transcription factors are bound. And then we can identify these methylated regions and these footprints by using Oxford Nanopore sequencing. So as I said, this technique has multiple different names, chromatin stenciling being one of them. But we have called this single molecule footprinting because the key thing we're looking for is these footprints where transcription factors are bound. So just a very quick laboratory overview. We take cells or pancreatic tissue, and we isolate the nuclei from these, and we incubate with our 2 different methyltransferases. We then extract the DNA, which still has our methylation and our transcription factors bound. And then we do methylation-sensitive Oxford Nanopore sequencing. So I'm not going to go into too much detail of the data processing, but we basically use all context 5MC and 6MA base calling using Dorado. And then we've developed a tool for processing these induced methylations, which allows us to identify the 5MC, but we can also distinctly identify the CG and the GC methylation, and then also our 6MA. So I wanted to now just kind of tell you about the amount of information that we can achieve with this method. So I've shown here this is a single nucleus attack sequencing data from pancreatic beta cell over a disease-relevant region. So this is a gene called FOXA2. And then this is the amount of information we can achieve with our method. So I've shown you here in blue is our GPC methylation, and then in orange is our 6-MA methylation. Each line being a read, and each of the colored boxes being our accessible or our methylated regions. And then in black, this is the average methylation over this region. So what's really great for us to see is that we're seeing these accessible regions which align with the accessible regions from the attack data. But also, we get this additional information in regions that traditional chromatin accessibility just wouldn't be able to achieve. So we can get a lot more information than we ever would have before. So if we were just wanting to look at the chromatin accessibility, we can still get enough data even from 15x coverage, but 30x obviously is ideal. So depth starts to become really, really important. So as you can see, we've got a lot of depth with the GPC. So this was achieved on a min-ion, but with adaptive sampling. Depth becomes really important when we want to start looking at the base pair level. So I'm going to use a CTCF site as an example for looking at this base pair resolution. Firstly, because we use these CTCF sites as a positive control for making sure our methylation has worked. But also these CTCF sites have a very characteristic accessibility pattern. So in the very middle is the CTCF transcription factor, which is bound. Immediately surrounding that are peaks of accessibility. But then surrounding the actual CTCF site are nucleosomes. And then we get this kind of wave pattern of accessibility, which correspond to the accessibility surrounding the nucleosomes. And we've identified this pattern in the 2 different methyltransferases that we use, but also in the different cell types that we've been looking at. But really, so this is all of our CTCF sites, an average of all the CTCF sites. The really exciting bit is when we start to look actually at a single CTCF site. So again, here is our GPC methylation in blue and our 6-MA methylation in orange. And right in the middle is where our transcription factor is bound, and that is where our footprint is. So you can see right in the middle is our CTCF footprint. So there's no methylation there, but surrounding that is very strong accessibility, very strong methylation, so a lot of accessibility in these regions. But what was also really interesting is that we can also see the accessible and non-accessible regions, which are the nucleosome footprints as well. So the blue being where it's accessible, the blue and orange accessible, and the where there's nothing, that's where the nucleosome is bound. So this type of resolution is just not possible with standard chromatin accessibility methods, and it's something that's particularly unique and really exciting for us to be able to identify. Of course, you can do this. I have just used CTCF sites as an example, but you can then go and look at your disease-relevant regions and look for these transcription factor binding. So to summarize, we can use base pair resolution to look at regulatory activity using Oxford Nanopore sequencing. We have a huge increase of information over the commonly used chromatin accessibility methods. We can do direct measurement of 2 exogenous DNA modifications, and we can provide information at the level of regulatory variants. So before I finish, I just kind of wanted to give you a bit of roadmap for our future. So at the moment, we are using cell types. So we're using endo-C-beta-H1 cells, which is an immortalized pancreatic beta cell. But we are also using differentiated pancreatic beta cell stem cells. We are also going to be using in vivo models. So we're going to be using isolated human islets, as well as human fetal pancreatic tissue across development. And then the idea is that we build this base pair level regulatory map of the developing and the adult human pancreas from a stem cell to an adult islet. And really, this resource will be really quite revolutionary for helping us understand the genetic susceptibility to diabetes. Again, important to note, we are particularly interested in diabetes and in the pancreas, but this can be translated to really any disease type. And we are now working with other teams to look at this in the brain. But we are also, if anyone would like to collaborate with us, we are also looking to collaborate on the application, the methodology, and any of the bioinformatics. So I would just like to thank Nick Owens and his group, and also a lot of other people at the University of Exeter for their support, and also to our funders. And thank you very much.